Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel Pharma World. Today's topic is prevention and control of nitrosamines in pharmaceutical products. Annex 2 of WHO Expert Committee on Specifications for Pharmaceutical Preparations 58th report provides a detailed strategy on identification and control of the nitrosamine impurities in the pharmaceutical products in WHO Technical Report Series number 1060 dated 2025. Let us understand the requirements in this video. The source of nitrosamines. N-nitrosamine, that is nitrosamines, are a group of compounds with a chemical structure R1N, R2NO, which are classified as probable human carcinogens. These nitrosamines are present in many SARTAN molecules, including valsartan. These impurities were subsequently detected in other medicines belonging to the SARTAN family, including N-nitrosodiethylamine NDEA, N-nitrosodiisopropylamine, that is NDIPA, N-nitrosoethyl isopropylamine, that is NEIPA, and N-nitroso N-methyl 4-aminobutyric acid, that is NMBA. WHO Technical Report Series number 1060 of 2025 prescribes several strategies for control of this impurity. This guide gives a comprehensive insight into the requirements. Let us understand in brief what this guideline prescribes. Nitrosamines can form by various pathways, including a nitrosation reaction between amines, secondary amines, and nitrosating agents originating from nitrite salts under acidic conditions. The source of these amines is from the solvents, reagents that are used in the process. They can be degradants or related substances from the API. Tertiary and quaternary amines may also be contaminated with secondary amines, either as impurities from the route of synthesis or as degradation products. Nitrating agents are generated from reagents like sodium nitrate, nitrous acid, nitric oxide, nitrosyl halides, dinitrogen trioxide, dinitrogen tetroxide, and other organic nitrates. There are also various sources of nitrosating agents such as recycled solvents or reused catalysts from different processes or across manufacturing lines with inadequate control and inappropriate monitoring. The risk for presence of nitrosamines. Some nitrosamines impurities may increase the risk of cancer in case of exposure above acceptable levels over long periods of time. Intake below acceptable limits in the entire lifetime is not expected to significantly increase cancer risk. Also, the actual health risk further will vary from person to person and depends on the nitrosamine impurity. The risk depends on several factors such as the daily dose of the medication, how long the medication is taken, the patient population, the level of nitrosamine impurity in the finished product, and cumulative exposure from multiple sources of nitrosamines. These factors decide on the risk of cancer. Some nitrosamines are highly potent mutagenic agents that have been classified as probable human carcinogens. Mutagenic impurities can cause changes or mutation in the genetic material of an organism. The control strategy. It is necessary to perform risk assessment to determine whether their products are at risk of containing nitrosamine impurities and ensure that the levels of impurities in the finished pharmaceutical product, that is FPP, do not exceed the acceptable limits. The risk assessment should include focus on premises, equipment, materials, route of synthesis, production process, and interactions between chemicals, excipients, solvents, APIs, and packaging components, as well as the dosage form and the intended use of the product. The potential presence of the above listed amines and nitrating reagents should be captured comprehensive way in the risk assessment report. The risk assessment should be done for all FPPs and should be reassessed, reviewed periodically. The guideline also prescribes that, in accordance with quality management principles, manufacturers should consider manufacturing changes and shifts over the product life cycle that may impact the potential for nitrosamine impurities, including new source of raw material or excipient, etc. Consideration should also be given to the product degradation and shelf life. WHO Technical Report Series number 981 Annexure 2 provides detailed path forward for effective risk management. This is very elaborate version of the quality risk management ICH Q9. The prescription uses all tools of ICH Q9, the FMECA tool for risk assessment. A detailed example on various categories with scoring is given in section 6 of this guide, TRS 681. The probability and impact metrics are well explained. List of nitrosamines. Nitrosamines impurities include N-nitroso-dimethylamine NDMA, N-nitroso-diethylamine NDEA, N-nitroso-diisopropylamine NDIPA, N-nitroso-N-methyl 4-amino-butyric acid NMBA, 1-methyl 4-nitroso-piperazine MNP, N-nitroso-ethyl isopropylamine NEIPA, and N-nitroso-dibutylamine NDBA. This is the list of potential nitrosamine impurities. If the risk assessment determines that there is no potential risk or harm relating to nitrosamine impurities, then there is no need to take further action. So it is important that the risk assessment report should be comprehensive enough to establish that there are no questions unanswered while determining the risk. All justifications should be scientifically valid with evidence. Where a risk relating to nitrosamine impurities is identified, confirmatory testing should be done using appropriately validated methods in accordance with current good practices. The analytical methods should be validated as per USP chapter 1225. It is necessary to conduct root cause analysis to determine the possible or probable cause of the formation of or contamination with nitrosamine impurities. The root cause analysis should be supplemental to the risk assessment. The detailed path forward for root cause analysis is provided in the next slide. Areas to look for in risk assessment: the root cause. The root cause could include amines and nitrate reactions. Formation of nitrosamines is possible in the presence of secondary amines and nitrite salts under acidic reaction conditions. Under such conditions, nitrite salts may form nitrous acid, which can react with an amine to form a nitrosamine. So this should be monitored for any potential source of nitrosamine impurities. Amine functional groups in processing. Amines may be present in the structures of some APIs and their starting materials, intermediates, related compounds, and degradants. It may be added as reagent or solvent or a catalyst in manufacturing processes. Secondary amines may react with nitrous acid or other nitrosating agents, leading to the formation of nitrosamine. Amide solvents are also a source of secondary amines to generate nitrosamines. Introduction of nitrosamine impurities. Starting materials and intermediates may be at risk for nitrosamine formation due to their structure and the manufacturing processes. This may also be at risk due to cross-contamination if they are manufactured at sites where nitrosamine impurities are formed during other processes. Sometimes it may be contaminated during storage, shipment, and distribution. Fresh or recovered raw materials, including solvents. Recovered solvents, reagents, and catalysts may also pose a risk of nitrosamine impurities due to the presence of some residual amines. Water. Water may also be a source of contamination as they may contain nitrates and other contaminants. Environmental air and contamination. Inlet air to equipment such as fluidized bed dryers may have some nitrosating agents, atmospheric nitrogen dioxide. Quenching process as a source of nitrosamine contamination. The use of nitrite or other nitrosating agents as quenching reagent could also be a potential source for nitrosamine contamination. All these aspects should be considered as root causes while carrying out the risk assessment. Other focus points. Risk control strategy should also include the following. In addition to the above, it is necessary to include the following for risk assessment: excipients and packaging materials. Impurities such as nitrite can be found in a range of commonly used excipients. Packaging materials like nitrocellulose in press-through pack aluminum printing ink and polyvinyl chloride PVC container material can contribute to nitrosamine impurities. Active pharmaceutical ingredients (APIs). The APIs may also have potential nitrosamine impurities to contribute. It is necessary for FPP manufacturer to request the API manufacturer to share any nitrosamine risks from purchasing their materials. This includes risk of nitrosamine and risk of presence of amines or nitrosating agents. Information on usage of recovered materials may also be requested. Finished pharmaceutical products (FPP) manufacturers. The risk assessment should include evaluation of the supply chain, excipients, API processing, utilities, storage, distribution, and degradation that could introduce nitrosamine during production or storage. Analytical methods. Pharmacopoeial or validated. In the case of non-pharmacopoeial analytical procedures should be used when testing for the presence of nitrosamines. The analytical method should be validated as per USP 1225 and ICH Q2R. If the compendial test method is utilized, it is necessary to verify the method as per USP 1226 when adopted first time in the laboratory. The procedure should be sufficiently sensitive for the determination of the specific nitrosamines in the product. Since the levels of nitrosamine are very low, it is necessary to establish the LOD and LOQ values at nanogram level. I hope that this brief insight into the subject is understood well. Read the TRS series 981 and 1060 texts for more detailed information. Thanks for watching. For more videos, please do subscribe, like and share. Also, please leave a message in the comments box for any further support. Thank you.